same complex will represent from the endogenous pathway at the effector level. So let me just briefly introduce to you the phagocytic system and where these things are going to happen. The phagosomes form by surrounding by the plasma membrane of a particle. This is an actin-dependent process, and then engulfed when there is fusion of these two pieces of plasma membrane, and engulfment occurs. And basically, the early phagosome is composed of plasma membrane. And then there is a membrane exchange process that starts as soon as the engulfment has occurred. And this exchange is first with early endosomes, and then with late intermediate multivesicular endosomes, and finally with lysosomes. And the result of all this membrane exchange between the endocytic pathway and the phagocytic pathway is that phagosomes mature, and they resemble first to early endosomes, then to late endosomes, and then to lysosomes. And they acquire through this exchange, and the mode of exchange actually has been described some years ago by Michel Desjardins and Gareth Griffiths, and they call this the kiss and run model. Basically what happens is that there are transient fusions between the two compartments which fuse and then separe, and a piece of endosome will remain on the phagosome side. And so this is the way how phagosomes acquire the protein. Yeah, that's interesting. Actually, I don't think there is any evidence that type 1 interferon regulates pH or phagosomal maturation, unlike maybe that's a question for Pierre that I see sitting there. It is, gamma interferon regulates phagosomal function strongly. Pierre, do you know if type 1 interference regulates? No? I don't think there is any evidence for that. It would be interesting to know.
And what about LPS onto the activity of NOx2 and...
Okay, what we see with LPS, so what we did for the moment is analyze the phagosome function in terms of pH by adding together the beads and LPS, but we didn't do it with beads which are having LPS bound to the beads, as Ruslan and other people do. And what we see is an increase in alkalinization. We never saw increased acidification in our hands by treating the cells with LPS. So there is more NOx2 activity and more alkalinization in the first hours.
Sebastian, brilliant story. I have a question on this lysosomal, this inhibitory lysosome-like vesicles. I suppose that if you say they are a particular subpopulation of lysosomes, that there are other lysosomes in dendritic cells that are different, right? So there are two populations of LAMP2 positive vesicles in dendritic cells. Now the question is, how are they different? Are, for instance, these inhibitory lysosomes non-acidic in nature, as you would expect, as opposed to the normal lysosomes? Do all lysosomes have the same composition in the antigenic makeup or the food?
Obviously, they did the experiment and they had very bad surprise. If you do that early, you might. Anyway, we can continue.
So maybe if you can take your seats again, we can continue on with this afternoon's session. So the next speaker is Klaus-Michael de Bertin from Ulm. The title of his presentation is Apoptosis Regulators in Cancer Therapy.
Yeah, I'd like to thank Laurence, Peter and Guido for bringing us together here, which is really a workshop because there are a lot of discussions, and because this is a workshop, I have actually decided to switch gears a little bit and talk rather about something that is going on in the lab and is mostly unpublished data. So it's pretty obvious that the cell death pathways, you can see that I'm not talking about apoptosis, but about cell death pathways have implications for disease, and of course the hope is, especially in cancer, that it provides tools and targets for cancer therapy. Now, our lab for many years, and this is just a summary of the most important topics of the past years, has been involved in trying to identify targets for therapeutic intervention, also from a clinical perspective, because I'm heading a clinical department, and we have done a lot of work in trying to sensitize tumor cells by various means, to use embryonic stem cell derived progenitor cells to bring therapeutic apoptosis-inducing principles into the tumor, and most recently worked in patients to monitor therapy response by apoptosis and molecular markers during therapy. But I will not talk about actually apoptosis in cancer. This is an immunology meeting, and I thought I would share with class 1 negative cell line was obtained from Wolfram Osen from the DKZ in Heidelberg. Thank you very much.
Thank you. It's time for question inhibitory studies, and I know some of you have seen this before, some of you haven't, and the young PhD students here shouldn't be put off when your supervisor, in this case Chris Haslett, blows up your model, because I showed him this model and he said it looked like just like a toilet down which apoptotic cells were being flushed into the macrophage, and this really upset me, and it set me off on a pathway of transfection studies and knockouts and all this to prove this toilet model conclusively, if you like. But at that time, what I want to get across was that we had a very simple idea of how dying cells were cleared. We thought they had on their surface injury signals which were recognised with very high fidelity, and bang, you were eaten. And what I hope I can show on the next slide, I might have to click on it, just click. So this is time-lapse video microscopy of macrophages interacting with neutrophils. And deliberately, it's a mixed population of neutrophils. Some of them are fairly healthy cells, and you can see these are the ones that are shape-changing and moving around. And some of them are apoptotic cells, which remain spherical. And what I want you to do is fix your attention up here, and we'll just click it again. Oh, pardon me. And I'll go back. Sorry, the software just didn't read across, sorry. Right, okay, so fix your attention on that apoptotic cell there. It's been captured by that zipper-like mechanism. It's been eaten, and here's another macrophage that's sort of got an apoptotic cell, and it's almost like a lion with an antelope. It's shaking it before it's eating it. But the fascinating thing is that while that one got eaten and destroyed, this one is still sitting on the surface, and there are other ones over here that are just rattling around being sampled by lots of macrophages, and there are healthy neutrophils being repelled from the surface of macrophages. And I don't want to make very much of that, other than to say that what that says really is that this process is much, much more complex than we ever thought it was. And of course the complexity of this has become more obvious over time as a large number of groups have added to the kind of molecular scenery of recognition of dying cells. So there are, if you like, innate recognition of non-self using systems like C1Q. There's recognition of altered self with all these kind of scavenger receptors. And then what Simon Brown and I have been working on for some time is, if you like, recognition of non-detaching self. And I want you to concentrate on this interaction that Simon discovered, which is a homophilic interaction between two immunoglobulin superfamily removal of dying cells. And again, I may be a bit out of date, but it looks to me as if there's like one receptor and some molecules involved in cytoskeletal reorganization, and that's enough to differentiate and grab dying cells and eat them. And of course, we've already seen that in the mammalian system, we've got much more complexity. And I believe that this complexity has evolved for a reason, and that is because there's added functions grafted on to waste disposal. And I just want to talk briefly about my favourite added function, which is suppression of inflammation. And to do that, I want to go back to the toilet model and focus.
Dear colleagues and friends, I'm happy to be here and participate for this meeting. I shall bring best wishes from the German-Russian cooperation for immunotherapy, and we have gained very good results. We have shown in mice and rat apoptosis in deminiature cells of liver, spleen, and brain cortex, and proliferation in miniature cells. And we have treated thousands of patients with good results, and these results have led to a patent which will be now used 07. And I'm happy to speak to you and announce that Freiburg Institute, Natasha Shalizova, Valery Lachenko from the Menshikov Medical Academy, and Professor Anisimov from the Oncological Department, and also from the Menshikov Medical Academy would like to join the ECTO. So it's a possibility to enlarge ECTO, and some of our members are already, some of these people are already members of your organization. And in 07 we will have a big meeting in St. Petersburg, and we keep you informed. And so I say have a nice day, and thank you for your attention.
We will now go and start with the session. We will first hear Richard Favell on kinase signaling cascade.
That every second, as it were, millions of cells are dying within us, yet we have the good sense not to make dramatic immune responses to these cells. And so the question I want to deal with is why that is, and what the underlying mechanisms that there are which play a role in this. And I will take the thesis that there is a very important mechanism which plays a role in this, and that is the cytokine TGF-beta. And you've heard some of this yesterday already. So TGF-beta, of course, is a cytokine that plays a role in pretty much all processes in the body. It's involved in oncogenesis, as you've heard yesterday. It's involved in important. They make IL-10 and TGF-beta, although in psoriasis commonly don't show this up. We know these are crucial because they prevent autoimmune disease under normal circumstances. If you delete them, you get disease. If you add them, you can block autoimmune diseases in a variety of models. So the question is then, how important?
They're surveying the tumour, and they're trying to decide whether a cell is a target and whether it should be killed. And if we look at this again a little closer, here we see a tumour cell, and here we see a cytotoxic lymphocyte. And you can see the CTL come over and recognize its target. And this is where we get interested in the whole situation. We want to know what's happened from this point onwards. And of course, there's two possibilities. There's two ways that a CTL can kill its target. And one of them is the trail and death receptor mediated mechanism, such as what Henning works on. But the one that we're interested in is this granule exocytosis mediated cell death. And what happens is, when the CTL comes in and makes a synapse with its target, essentially the granules, the cytotoxic granules, move from the back of the cell to the front of the cell. They become polarized. And then granzymes and perforin, so perforin here by these large circles in red, somehow deliver granzymes, the smaller circles, to the target cell. And these various granzymes can induce various death pathways by cleaving various substrates and cause death of the target cell. So the story becomes a little complex at this stage, because when we put in cytotoxic drugs, we sort of think that the pathway is set out from a fact of the cytotoxic stress induces activation of a BH3 family member, let's say mitochondrial activation, you get caspases, and then you get death. And things mightn't obviously be that simple with the difference between immunogenic and non-immunogenic death. But in the cytotoxic granules, we have multiple granzymes that can induce various deaths, such as apoptosis and non-apoptotic cell death. We have other granzymes that we don't know what's happening at all, other granule components that we don't know what's happening at all, and we know that perforin alone can induce a lytic death if delivered at high enough concentrations, and granulysin can also induce lysis. So this gives us a situation where we don't really know what a CTL is doing to kill its target.
Glad to meet you. Hello. Hello. You're the president of the ECTO. Please tell some words about the organization. What is the purpose about it?
Well, ACTA was founded now 14 years ago, and started with a series of Europe conferences on apoptosis, a topic that became recognized in the early 90s as a major form by which cells are dying, with widespread implication for many diseases, including cancer, including autoimmunity, including hypoxic cell death, cardiac stroke and brain stroke. In the 90s, this field really has exploded in basic research because the signaling systems by which cells are told to die have been discovered. And this field is now entering areas such as the immune system, how the immune system deals with cell death and dying cells, and this is the topic of this particular conference. The European Cell Death Organization has about 300 members throughout Europe. All the larger research institutes and some of the major clinics participate in this group, and we have regular meetings every year of this European Cell Death Organization and some workshops like the one that is now held here in Freiburg.
So I gave a little greeting address from St. Petersburg and Moscow and from the German-Russian cooperation on immunotherapy. They have asked there for a cooperation with you, your organization of course, and I would like to hear your opinion about it to enlarge ACTA also to the Russian countries and so on. What do you think about it?
I think we already had in the past participants from Russian countries, and actually we even had planned to have one of these meetings in St. Petersburg.
Good.
And we certainly welcome also all the members, researchers from Russia that want to participate in this enterprise. There is no restriction to become an actual member. It's open for all European countries.
Okay, so we are on the same line. Thank you for the interview. Okay.
We have not tested that yet because we could not get access to the drug. What we have done is to check on our. We don't have cells treated with even with the medicine, so I cannot answer.
Invite me to share some data about these very special T cells. I think we're far away from the death, but I think, and still an hypothesis, that these very funny T cells may be important to sense the gut microbial environment. So I will try to first describe what are those T cells and to show also some preliminary data about the influence of the gut microbial environment about their accumulation in the gut. As you know, the classical T cell recognizes multi-peptide complexes on the ABC as a class 1 or class 2 of this peptide, and it means that you have a lot of different T cells, different PCR, which represent a very large numbers of different multi-peptide complexes. On the opposite end, you have the innate cells, which are a few conserved receptors.
Possibility to kill tumor cells. So before talking about IKDC, I just want to try to summarize some important problems we have to deal with in cancer therapy, which are the hallmarks of cancer itself. These are its self-sufficiency in growth signals, its insensitivity to anti-growth signals, resistance to apoptosis, its great replicative potential, sustained angiogenesis, tissue invasion, metastasis. And as 7th point, for sure, the